Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Novartis, Allergan, Janssen, Biogen, AGTC, MeiraGTx, and the SickKids Foundation. We'd also like to thank Accessible Media Inc., our national accessibility partner. Everyone should download the AMI app, which is a fabulous resource, and you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below. Thank you again for joining us.
Morgan, Lister, thank you so much for having me today. I really, really appreciate it. To all of you who are tuning in, including my mother, hi Mom, hi Stan. It's an absolute pleasure to be here. I always really enjoy working with Fighting Blindness Canada. I've attended their Vision Quest a number of times over the years. And if you're part of the community and you haven't attended Vision Quest, it's an event I highly, highly recommend. It's very fun to interact with the patients and interact with the donors and other scientists and clinicians. It's a really great event and I really appreciate it. So the talk I'm going to give today, I'll share my screen in a moment, is a bit of a repeat of the talk I gave in Calgary in 2019 in October. And because it's a little bit of a repeat of the talk, I'm going to repeat the story that I tell in the beginning too. So I mean, whenever I get ready to give a talk, I always get a little nervous. So I like to tell a little story, tell a little joke. So you guys get a little relaxed, I get a little relaxed. And so when I first moved to Alberta to pursue my postdoctoral fellowship, I got linked up with a new hotshot master's student who was a really, really intelligent, really smart guy. And him and I were driving around Edmonton together. We'd become fast friends. And we saw an individual who had a visual impairment with their cane walking down the road. And he looked at me and said, you know, I wish I could, not while he was driving, for the record. He looked at me and he said, you know, I wish I could get so good at genetics that I could look at somebody and just know what disease they had and maybe like what gene might be causing their condition. And I said, oh, he has, I'm pretty sure, X-linked retinitis pigmentosa. And, you know, the student looks at me and kind of like, how could you possibly know that? There's no way you've gotten good enough at genetics that you could possibly know that. And I went, oh, no, that's Jerry. I met him at Vision Quest last year. So that's a story I really like to tell out to start things off. I hope you guys got a little chuckle out of that. I know I did. Jerry, if you're in the crowd, I'm sorry for picking on you again. So we're going to start off my talk here. So today I'm going to be talking about genetics and cell models of eye diseases. Again, as Morgan so kindly pointed out, my name is Lance Doucette. I'm at the University of Alberta. I'm a research associate. I am a PhD scientist. That is to say I'm a research scientist. I am not a medical doctor and I'm not a clinician. So anything that I say should not be interpreted as medical advice. And I come from the Department of Ophthalmology and Visual Sciences. And, oh, here we go. So, come on, change. There we go. So I work with this fellow here in the top left corner. His name is Dr. Ian McDonald. If you're from Alberta, you probably know his name or have probably seen him, as he's one of the leading people here in Alberta in terms of both genetics and ophthalmology. He's also the leader of Canada's first gene therapy trial in choroideremia, which just clued up a little while ago. And he's a great, really nice guy. If you ever see him at conferences or at Vision Quests, go up, say hello, tell him Lance sent you. He'd get a kick out of that. So the purpose of my talk today is going to be to inform you about what we're doing to study visual diseases, particularly in Dr. McDonald's lab, the work that I do, and then walk you through some of the studies that we do, how we do them, and ultimately what they may mean to you as a patient or to the scientific community at large. Then we're going to talk a little bit briefly about where we want to go with these studies and what we're hoping to achieve. I'm going to do another little quick plug here, and that's for the Alberta Vision Network. In Edmonton, we are lucky to have if not the largest, then very close concentration of vision scientists in Canada. And this is a group originally founded by Dr. Mike Walter and Dr. Orden Lehman. And we're a group devoted to the advancement of visual sciences and therapies in Alberta. And we bring in all the visual scientists from particularly Edmonton and Calgary, but we also have collaboraters in Lethbridge and Red Deer and all about. In this group, we have clinicians, geneticists, cell biologists, animal scientists, including mice, rats, zebrafish, and we conduct monthly meetings, which we've been falling a bit down on with the COVID-19 crisis. And we do have an annual research day where we highlight all our trainees. So we train PhD and master's students and postdocs to do research, and we highlight all of the work that's been done, and we bring in national and international speakers to educate us on what they're up to. And the little cute picture here below is what you see is the photoreceptor mosaic that you see in zebrafish. So photoreceptors, the cells at the back of the eye that are responsible for picking up light and translating that light into vision. Another little note on my talk, I realize many individuals are likely visually impaired. So most of these are just notes for me to talk about. If there is something that will come up on the screen that you're not able to see, I will try my best to describe it in great detail. So the thing I'm going to start off talking about today is, of course, the genetics of inherited eye diseases, the thing that I'm most passionate about studying. Our studies all aim to find genes that cause inherited diseases of the eye. The Human Genome Project was completed in 2002, so medical genetics itself is a very, very young science, as we've only ever determined for the past 18 years what genes are out there, and now determining how mutations in genes might be tied to disease is an ongoing battle that we're all still trying to figure out day to day. So in our lab, and mostly what I do, is to try to determine new genes. We're looking for novel associations or new associations with retinal diseases or other ocular diseases. And we do this in consultation with clinicians like Dr. Ian McDonald or Dr. Orden Lehman, or perhaps the ophthalmologist that you see. And the ophthalmologist at this point will order genetic tests for commonly known diseases. So if you have retinitis pigmentosa, a condition which affects the rod photoreceptors, you'll be sent for a panel that will test for genes that are known to cause retinitis pigmentosa. But if these come back negative, that's when your file lands on my desk, and we're starting to look for new genes that will help inform future therapies and help kind of expand our broad base of what genetics are out there for retinal diseases. When your file comes across my desk, I work with Dr. McDonald to assess the likelihood of identifying a new gene. And this is dependent on, how many family members we have available, what's your family history, how much information do we have about you. And we use this information to determine if this would be a good place to start our research studies. And the end goal is accomplished through something called next generation sequencing. So I'll explain a little bit about that a little later, but just keep that word in mind. Because before we talk about that, we want to talk about very, very basic genetics. In the book that is the human genome, there are only 4 letters. There's A, G, T, and C. And those are the 4 letters, just like our alphabet that make up English words. Those are the 4 letters that make up the book of the human. And it's the sequences of these letters that tell a cell which protein to make, and proteins are small molecules that make up the structural parts of your muscles. They might be enzymes that break down fats or help break down the food that you eat. So there are 2, the structural and the enzymes that are made by genes. It's the sequence of those 4 letters that tell a cell when to make a protein, how to make a protein, and when to stop making protein. So for the purpose of this talk, we're going to call a gene a sequence of DNA that makes a protein. There are other sequences that are in between genes. These are called intergenic sequences. This is the same as you'd call something international, intergenic, in between genes. We don't know a whole lot about these intergenic sequences. We're learning more about them as we go, but we kind of get the sense that they're a thermostat. So instead of turning a gene on or off like a flick of a switch, these things act as thermostats and say, okay, we'll turn the gene up a little tiny bit and then turn it back down when it's time, or turn it all the way up and then leave it at middle for good maintenance, that nice 20-degree mark. But we're not going to talk too much about those. We're mostly going to be focusing on the parts of the genome that make a protein, or genes. So when you go to see your clinician or your doctor, you might be asked to provide a DNA sample. And I want to give you a sense now of what we do once we have that sample in hand. Once we have your family history, your clinical information, the first thing we talk about is in inherited retinal diseases. These are the ones that are inherited through families, or we call Mendelian inheritance, after the fellow who discovered it. This is the pea plant guy, if you've ever heard about him, Gregor Mendel. First, we start off determining the mode of inheritance. So there are 3 modes of inheritance that we tend to lean towards first, and these are dominant, recessive, and X-linked. I'll go through each one very briefly. So for dominant, you have 2 sets of chromosomes in your body, and in a dominant condition, you would have one of these chromosomes that's got a mutation on it, and the other one is normal. So if you have the condition, you have the mutation on one chromosome, you have a 50% chance of giving that one chromosome, that mutated chromosome, to your child, and a 50% chance of giving that normal chromosome to your child. So in a big extended family tree, what you end up seeing is probably about 50% of children have this dominant disease from somebody who already has the condition. So an affected parent giving birth to 50% affected kids. This is called a dominant condition. We'd see the condition through each generation of the family tree. The other thing to consider is recessive. Typically, this looks like unaffected parents with affected children. It almost looks like it skips a generation in family trees. And the reason for this is because in the previous case, we said that you need to have one mutated chromosome to have the condition. In recessive conditions, you need to have two copies. So you would have two parents who have a mutated copy each, but this is not enough to bring about the disease. They'll call it carriers. And when they breed, they each have a 50% chance of donating their mutant chromosome to a child. So 50% from mom, 50% from dad, and that leaves a 25% chance of having an affected child. So what it looks like, again, on these big extended family trees is you might look like it skips generations. The third one we like to consider is X-linked. As we might know, to be a male, you require an X chromosome and a Y chromosome to be a male. To be a female, you require two X chromosomes. So in these cases, the disease gene is on the X chromosome. So what you end up seeing in these families, in the big extended family trees, is you see no male-to-male transmission, because in order for a male to have a male, he has to give his Y chromosome. The X chromosome comes from mom. So you have carrier females giving birth to affected, typically boys, because boys only have one X chromosome. That if they get the mutant X chromosome, it expresses the disease, whereas in females, you have two X chromosomes. You might have one bad one and one good one, and often the good one is enough to make the disease not appear. So these are the three things we try to consider when given your family history. And once we kind of get this all figured out, we move on to this next generation sequencing. So next generation sequencing is a technology that came about probably in the last 10 to 15 years. It's new-ish, and it allows us to get all those letters of the human book, that A, G, T, and C, every sequence in the human genome we can get with this technology. And it's done very cheaply at this point. We can do it for about, I think it's about 1,200 Canadian a sample, probably a little bit more right now. So it's gotten very, very cheap, considering it started at about 10 to 15,000 a sample. It's come down a long way. One form of this is called exome sequencing. So don't worry too much about the terminology here. We just call it WES, W-E-S. And what this technology allows us to do is it allows us to look at the part of the genome that is just the genes, just the parts that make up the proteins. And we know from previous studies and from the Human Genome Project that there are about 23,000 genes in the human genome, but that only accounts for about 2% of the whole human book. So 98% is that intergenic sequence I was talking about, the stuff that provides the thermostat, kind of turning the genes up and down when needed. The actual genes that control the protein sequences are only about 2%, but it's where most of the mutations are found. Using whole exome sequencing, or WES, we can determine typically about 50 to 60% of patients using this. The pros of it are, of course, it's very cheap. Exome sequencing, as opposed to the whole genome, is about $600 or so. But the cons are, because we're only looking at 2% of the genome, sometimes we miss things. Like I said, we only get about 50 to 60% of patients with WES, so that means we're missing about 40 to 50% of patients at the end of the day who might have variations or mutations in that other 98% of the genome. So in every individual, you have your genome, you have your book, your human book. Every individual has about 4 to 5 million variations. And when we say variations, we mean from a reference genome that was determined from the Human Genome Project, just kind of what we call normal. But we're all normal in our own way. These 4 to 5 million variations that exist in individuals give us the differences between us. They determine why you're a little bit taller than your brother, or why your nose is a little smaller than your sister, or why you look so much different than the neighbor. These are the variations that give you the unique characteristics that are you. And many of these are called polymorphisms. It just means that they're different forms of the same gene that give rise to different looks, different characteristics. So an average whole exome sequencing file, when I get it back from your sequencing, has about 20,000 of these variations in it. So remember, we're only getting 2% of the whole genome instead of that whole 100%. So we get a lot less, which makes it a lot easier to analyze. But the problem is, how do we distinguish which of these polymorphisms or variations are causing your ocular disease and which ones are controlling, again, how big your nose are, how much hair you have or don't have in my case, and really determining which one is what we call the causative variation. So we do this through a number of different ways. The first is to get rid of any common ones, because if they're common, if it was a common mutation causing a retinal disease, everybody would have the retinal disease, but that's not the case. Right? So we can get rid of the common ones because there are a number of databases out there, such as the Human Genome Project, the 1,000 Genomes Project, the 100,000 Genomes Project, that this is entirely what they're built for. They get every letter in the human book and they determine how likely is that letter to be a different one. How often is that going to be mutated? So we can kind of toss out all the common variations and say, okay, that can't be it, because if this was it, everybody would have that disease. So we can throw those out. And that generally kicks out, you know, if we have these 20,000, that probably kicks out about 15,000 to 16,000 of these variations, because we have so many in an individual. We also rely on observations from other scientists, whether they're published on these databases or published in individual journals and papers that we then read. Maybe we find a rare variation in a person, but we find out that it actually has nothing to do with your disease. Then that's still important information that we have to let other scientists know. And we use that information to help determine what gene, what variation in that gene is causing your condition. The other thing we do, and probably the most important and most effective, is called segregation analysis, or put very simply, family comparison. If you have a condition and your brother doesn't, you guys share 50% of your DNA. So there's something in that 50% difference that's causing yours and not your brother's. So it's important, and I'll kind of put this out to all the patients, when you're coming in to get your genetic studies done, if you agree to be part of a research program, what makes our job the easiest and helps us get an answer to you the quickest is if you speak to your family members about coming into the research program yourselves. More people equals more power for our studies and allows us to get back to you in a more timely manner and really figure this out. Because a lot of the times we'll end up with one, maybe two people, and then we end up with, you know, probably 30 different genes that we're looking at, 30 different variations, and go, well, we can't tell which one it is. If we had an additional family member, that would help us determine it. So I implore you, speak to your family members, but don't be too pushy. Some people don't want to be part of research studies, and that's perfectly okay. Okay, so once you've convinced your whole family to come to our clinic and we draw your blood and we get your DNA and we do all this sequencing, we filter down that long list of 20,000 variations to a short list. So say now we have 24, or maybe even a dozen genes and variations that we think might be causing your condition by comparison to these databases, by comparison to the literature from other scientists, by associating with it your affected and unaffected family members. So we have a little bit of a short list at this point. The first thing we have to ask ourselves is, does your disease, on our short list of genes, are any of these genes associated with disease before? For example, if you have retinitis pigmentosa, which we talked about earlier, as it affects the rod photoreceptors, which are on the outside of your retina, and leave you with a lack of peripheral vision, so out here, and a lack of night vision as well. We'll ask, are any of the genes that we found variations, are any of those associated with retinitis pigmentosa? If so, we're probably off to a better start of your study and figuring out what gene is causing it. It's not new, but it might have been missed on the initial screens that we did when you saw your clinician the first time. The hardest part of it is that still there's a lot of genes that we simply just don't know enough about them. We might know what they do, but we might not know where they're turned on in the body. For example, if you have a gene that controls enzymes in your stomach for digesting food, you wouldn't expect those enzymes to be turned on in your retina, in your eye. But we need to kind of, and the problem is we don't understand a lot of these genes. There are many genes out there that haven't been studied well at all, and we know kind of what their function is, but we don't know where they're turned on or where they're turned off, or we might know where it's turned on, but we might not know what the function is. So we need to ask ourselves these questions when looking at our shortlist. Are any of these expressed in the retina, which means is it turned on in the eye? Or again, if we find something that's controlling, if we know that the gene is controlling the enzymes in your stomach that help you break down food, well, we know we can get rid of that one because it's not in the eye. So once we have our short list, we can kind of figure things out. And I mentioned earlier, sometimes we do the comparison of genes to a list of known genes, which will give us more information. I'd like to give you a little example of how this can play out. So we had a family come into Dr. McDonald's clinic, and they had what we conceived to be non-syndromic retinitis pigmentosa. So just the eye phenotype that we talked about, the loss of the rod photoreceptors, the night vision problems, the peripheral vision problems. And this whole story is an example of how the genetics really affected the medicine. Because they came in, remember we talked earlier about the modes of inheritance. We sat down with the modes of inheritance and we decided that they had 2 unaffected parents, 2 affected boys, and an unaffected sister. So what we thought was that the mother is a carrier of a mutation on her X chromosome, which she gave to the 2 boys, and that the sister was unaffected because she either didn't get that X chromosome or she had a spare good one to make up for it, that she was also a carrier. So we call this the X-linked form of inheritance. We know that there's only 3 genes that cause X-linked retinitis pigmentosa. It's RPGR, RP1, and RP2. And so we did a screen for those 3 genes right straight out of the clinic, and that came back negative. So that file came across my desk. Myself and Dr. McDonald discussed it, and we put this into the whole exome sequencing. So we got the whole exome sequencing back on the 2 affected boys and the affected sister and the mom and the dad. And we did the whole comparison. We said, okay, well, which ones are the common variations that might account for differences in height and weight and facial structure? We get rid of all those first. Then we do the data. Then we do the comparisons to known genes. And the first thing that fell out of this was a mutation in a gene called BBS1. BBS1 is really interesting because it does cause retinitis pigmentosa. It does cause that rod-dominated disease, but it causes other systemic problems too. So it can cause things like learning disabilities, obesity, extra toes or fingers, kidney problems, because of the certain structure it controls in the cell. So when we got this information back, we realized that we had been wrong about the initial diagnosis, that they didn't have non-syndromic retinitis pigmentosa. They had this broader condition called Bardet-Biedl syndrome. And when we went back to the family and told them, hey, we think you have mutation, we know you have mutations in BBS1 that probably explain this, the patients then told us, well, our son actually had an extra toe when he was a kid and had removed. And it just didn't occur to the patients at the time because naturally, what does my kid's extra toe have to do with his eye? Is something that you wouldn't even think about, but realized later that it was actually part of the disease. So I guess the other little piece of advice I want to give to the patients is when you do go see your clinicians, your ophthalmologists, make sure you communicate some of this extra information. Like if you had an extra toe, if you had a different condition as well when you were younger, communicate that as well, because this information becomes this information becomes really important when geneticists are dealing with it, because sometimes it's not just the eye, it's a whole body problem. The next example is an example of when we were looking for a new gene. We compared it to the lists. This was a case we had that had something called early onset drusen. So drusen are small little yellow deposits that exist in the back of the retina, and they tend to cluster around the center of the eye called the macula. And the macula is where you get most of your high vision, your high acuity vision. So if you're looking straight at something, you can see it really clearly. That's because of your macula. These deposits are associated with a form of macular degeneration called dry macular degeneration or age-related macular degeneration, which you guys may have heard about before. We did a normal genetic screen, which came back with nothing. This came across my desk. We discussed it with Dr. McDonald, said, yep, this looks good. Let's go ahead with it. So we did the whole exome sequencing. We did all those comparisons to the databases, to the known science, to the known genes, and nothing fell out. So from that, we got our short list, and we started asking questions about, well, which genes are turned on in the eye? Which genes would explain what's going on here in the eye? So we identified mutations in a gene, it's a really big gene called LRP1. And the reason we got really interested in this is because this thing is a janitor, effectively. So there's space between cells called the extracellular matrix. And what this protein does is it reaches out into that space between the cells and it cleans up garbage that might be deposited there, either through blood flow past the cells or other cells getting rid of stuff or just weird interactions with other cells. It would reach out, grab this garbage, pull it into the cell and degrade it and just get rid of it. The other reason we thought this was really interesting is because it was expressed, this thing is turned on in the exact spot where these deposits form. And they form under a layer called the retinal pigment epithelium. If you look at the picture of a retina, it's always red. That's because you get this picture of the blood vessels for one, but you're also getting pictures of pigment from the retinal pigment epithelium, which is at the far, far back of the eye. These deposits kind of sneak underneath the retinal pigment epithelium, and you can see them as yellow spots when you look at the retina. But this is exactly where LRP1 is turned on, at the back of the eye. So what we're hypothesizing is that as blood flows along the back of the retina, the back of the eye, it deposits stuff, cellular garbage, stuff that's in your blood, and LRP1 is responsible for taking that into the cell and degrading it and getting rid of it. We also noted that it interacts with a couple of components of these subretinal deposits. So these deposits are comprised of things like fats, cholesterol, immune proteins, and we realized that this thing actually can interact with fats, cholesterol, immune proteins. So it's turned on at the right spot, it does the right job, and it interacts with things that we know are part of the disease. So what we kind of assume is happening here is that the blood flows along the back of the retina, deposits this stuff, but the mutations in it make it either not able to bind to the right stuff, not able to pick up the garbage, or maybe it picks up the garbage a little bit more slowly and causes this stuff to accumulate, which leads to these small spots at the back of the eye. That's just kind of our working hypothesis right now. So, for example, for that one, now what? We have a gene. What else do we do with it? How do we prove that our gene is correct? How do we prove that we're on the right track? So in our lab, we use cell models. So cells are great ways to study the proteins that get mutated through gene variation. So remember, a gene is that sequence of those 4 letters, A, G, T, and C, that tell us how to make a protein. When you change those letters, that changes how the protein is made. Maybe it changes how much is made. Maybe it changes how the protein works. So what we can do with cells is we can ask the cell to make a lot of this protein, and then ask another set of cells to make a lot of the protein with the mutation in it. And then we can compare the function of these two things. So say if we were looking at a protein that was meant to break down fats, we could take some of that enzyme, make it in the cells, and then mix it with some fats, see how it breaks it down, and then compare it how it breaks it down compared to a mutation in that protein. So I hope that makes sense. The other thing we can do in cells is we can use gene editing in these new technologies, which many of you may have heard of, called CRISPR, which are proteins that can actually edit DNA in very specific ways in exactly the way we want them to happen. So we can introduce the mutation we find in you into a completely extraneous cell in order to better understand how that mutation affects the metabolism of the cell, how it affects the structure of the protein, how it influences the function of that protein. And we can also use cells to screen drugs. So we ask the cell, okay, make some of this mutant protein, and then we can use drugs to try to restore function to that protein by maybe refolding it a different way, or by restoring function, or maybe turning on another pathway that is related to that protein, but is not necessarily involved with that one protein. There is a cell line out there called HeLa, H-E-L-A. It is probably the most well-known cell model that's out there. This is called an immortal line. That is, it was discovered in 1952, and we have been growing it ever since. It was taken from an African-American lady in Philadelphia by a doctor named George Gay. There is a really, really wonderful book out there called The Immortal Life of Henrietta Lacks, and that's where the name HeLa comes from, Henrietta Lacks. And the story about how the cells were established, who they were taken from, how it affected her family, and some of the morals and ethics that surround things like when you give tissue to a research lab, who owns it? Or if you get a biopsy done, who owns that? But of course, this came from a cervical cancer that this poor lady had. And even though it is a very useful model for studying how the mutations might affect a protein, it's a cervical cancer cell. It's not an eye cell. So it's not really perfectly suited to doing these studies. It just gives us a good idea of how the mutation might affect the protein. So when you come to see your ophthalmologist or your geneticist, you might be asked for tissue so that we can obtain cells to bring back into the lab. Either that might be skin or blood. The skin cells in particular come from a small skin punch that you get a little anesthetic for, and then they take a small punch. We bring that into the lab and streak it out onto a plate. The cells grow from that little streak, and they grow for a long time. We can probably keep them for a number of years, and it gives us access to DNA later on down the road, so we don't have to recontact you to get more DNA for further studies. But those skin cells also contain everything about you in terms of your human book, your genome. That includes the mutations that control your eye disease, but also the other mutations that we talked about, the common mutations. The neat thing about doing drug studies, for example, in the other cells, the immortal cells, is that we can get an idea of what's happening with that mutant protein, but it completely ignores the rest of your genome. If we study it on actual cells that came from you, we can get a better idea about how your body might react given these new drugs as part of these research studies, right? So it makes it a little bit more accurate because all that information that makes you is there. But once again, as we did talk about in the immortal cells, skin cells are not eye cells. And they're not particularly well suited studies. There are some genes that are turned on in skin cells that are not turned on in the eye, and vice versa. They can give us a good idea, but they're not perfect. So next, what we might do is take some of those skin cells. And what's really, really exciting is some of the work that I've done in Dr. McDonald's lab is we can take those skin cells and turn them into stem cells. Now, stem cells are the cells that when you're first being made as an embryo, these cells can be turned into anything else. These cells need to know where to go, what to become, but they have the promise, the power to become any other cell in the body. So what we're able to do now with some really advanced tech is take your skin cells that come from you, turn them back into stem cells, and then hopefully turn them into a cell that makes more sense for your disease. So obviously for us, we want to take your skin, turn it back into a stem cell, then give it the correct program to turn back into a retinal cell. For our macular degeneration study, this is really, really important. So I'm going to show you a picture of what the stem cells look like. So what you're seeing on your screen now on the left is on the periphery, on the whole outside, these are cells in a dish. The cells on the outer periphery, they're very long, very tapered cells. These are cells from a skin biopsy that we took. Towards the center, you see this defined border of cells kind of sitting in the middle, and these cells are much, much smaller. They're not tapered. They're very, very tiny, and they're starting to get dark in their centers. These are stem cells. So what we did was we introduced certain factors into these skin cells, grew them in an environment conducive to creating stem cells, and these little colonies form. And we pick these colonies, we move them over into new dishes, and we expand them. So here on the right, you are again seeing an expanded colony with these small little dotted cells. All the little small black dots towards the center are all cells. And then you can see on the periphery, it forms these really, really interesting barriers. These, not barriers, what's the word I'm looking for? Boundaries, borders that look really, really good. So these are stem cells that we created from a human patient that we then wanted to turn into another cell. So this is a closer up version of these stem cells here on the left. If you can see this, what you're noticing is that they look very, very big. They have these black dots in the center of them. These are the nuclei or the nucleus, the center of the cell where the DNA is contained. And they're really, really big cells when you get up close. What we wanted to do was take these and turn them into the cells at the back of the eye, the ones that have that pigment, the retinal pigment epithelium. And we've been able to successfully do that. So on the right, what you're seeing is more of a cobblestone appearance. So kind of if you take a bunch of rocks and fit them together, it forms a nice looking pattern. And that's exactly what these cells, the retinal pigment epithelium cells, end up looking like once you've differentiated them. So now we have a cell from the patient that contains all the genetic information of that patient and is an actual eye retinal cell. So this is a little bit more well suited than the skin cells were, than the immortal cells were, to actually understanding how the gene mutations in these cells affect ocular health. So that's kind of going to do it for me, just as a concluding remark, a kind of a summary of everything we talked about today. When you come into the office to talk to your ophthalmologist, if we're looking for genes, we'll do the next generation sequencing. We'll filter down that list of genes to figure out what variation might be. We might ask you to bring in some more of your family members if possible. We might ask you for a biological sample, either skin or blood, or perhaps spit swabs, depending on what we need. And from this information, we can discover new genes in terms of a research basis. And the reason it's important is because we need to have an expanded idea of how the retina works, how when we mutate these genes, the retina gets sick. And this information can lead to new trials, new drugs. But I'm going to communicate that research is slow. It's slow because we have to make sure we're asking the right questions, that we're using the right cells, we're using the right model to study these questions. And ultimately, at the end of the day, we want you to be safe. So if we develop a therapy that comes from this research, we need to test and test and test and test and test to make sure that it's both efficient but safe for you. In terms of once we get the genetic idea, we can bring it back to the cells and the stem cells. Stem cells are great models for basic research into the conditions and for therapeutic assessments, but of course are not the end all be all because they're still cells, individual cells in a whole organ. So you got to keep in mind that the eye is comprised of many, 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 many different types of cells, and we're only looking at one. So stem cell models are getting a little better. We're able to create small little retinas in the dish that contain all those cells available in the retina. So the photoreceptors, any neurons that are there, are all in these mini retinas, and these are called organoids. This is kind of new cutting edge technology in the last 5 to 6 years, and it's not really caught on for everybody yet, but it's certainly coming along, and it's really, really exciting. But of course, animal models are still the cornerstone of any of this research. We have a cell, we figure out what the protein, what the gene mutation does. We should move this into a model where we have a whole organ. Like I said, we're only studying one cell. We need to study them all. And that ultimately comes down to models like mice or zebrafish or larger primates and that kind of research. So I hope you folks learned something today, and I'd be happy to take any questions that you might have.
Awesome. Thank you so much.
My pleasure.
We have a bunch of questions already that have come in through email, and of course the Q&A box is available to everyone. And if you'd like to ask a question over audio, you need to just press a couple keys on your keyboard, which will raise your hand, and then I'll know that you want to ask a question that way. So if you're on a PC, you press Alt-Y, and if you're on a Mac, you press Option-Y. So if anyone's interested in that, go ahead and do it, and I will be able to call on you in a minute. But let's kick it off with something we had from the email. So Gary sent in the question, I am a 60-year-old male with RP. My wife does not have RP. My 30-year-old daughter does not have RP, and her husband does not have RP. Their 28-year-old son and his wife both also do not have RP. Whose children may get RP sooner, my daughters or my sons?
Okay, so I'm actually going to share my screen again because I saw this question in the email earlier and I anticipated it. So Gary, if you're in the chat, this is what a normal, what a pedigree would look like. So squares are men, circles are females, and then if you go across left to right, that's a marriage, and then going down is children. So I hope you can see this. I hope I've made it big enough. If not, I do apologize, and I'll try to describe in greatest detail that I can. So Gary, this would be you here, the black square, and your unaffected wife, the circle, the open circle. And you have two children, one son, square, who is unaffected, and one daughter who's unaffected. So the best answer I can give you to your question as to whose is going to develop RP is, unless you know the mode of inheritance, like we talked about earlier, whether it's X-linked or dominant or recessive, we can't really be sure. But my guess would be that we're either talking about X-linked or recessive. So in the case of X-linked, I'm going to try to use a nice big, there we go. So Gary would have an X chromosome and a Y chromosome, and Gary's wife would have two X's. So Gary, if this X chromosome that you have here, this guy, has a mutation on it, draw that little star there, you have a 50% chance of giving that X chromosome to your daughter, when you have a child, you have a 50% chance of giving an X, which would make a girl, or a 50% chance of having a giving on the Y, which would make a boy. So in this case, you gave your Y chromosome to your boy. And in the other case, you gave your X chromosome to your girl. So in a case where your gene is X-linked, I don't have a pen, so you're going to have to bear with my terrible crude drawings here. In a case where you have X-linked disease, you potentially gave the X chromosome to your daughter, which would make her a carrier. So typically we put a dot in the middle of a carrier. But that's not for sure, right? We don't know what form of the disease you might have. In the case where you are in the case where we're talking about recessive, oops, my knee's too big, but that's okay. In the case where we're talking about things like recessive conditions, you would have, say, let's just say it's on your first chromosome. You would have chromosome 1 and chromosome 1, and say this one on the left is the one that has a mutation on it. You would have a 50% chance of passing that on to your son, or a 50% chance of donating it to your daughter, which would, again, make them carriers. And in the case of recessive, it's going to depend on the spouse. Maybe the spouse is a carrier for the same gene, then you would have a risk of having affected children. But so I should have said this at the beginning. Again, and I said at the beginning of my lecture that I'm a PhD, not an MD. Nothing I say should be interpreted as medical advice. I carry this big shield with me called the I am not a MD shield, and I'm going to hide behind that most of the questions. But that's how that would work. You have potential of having a couple of carriers, but it would depend on the carrier status of the spouse at that time. So without knowing exactly what form of the condition that you have, it would be impossible to tell. But those are the two kind of possibilities.
Okay, thank you. Very love the drawings. Those were for mouse drawing. Better than I could do, that's for sure. Okay, so another question we had from email, Khalid asks, let's just read through here. So it said when they were going through genetic counseling after receiving a diagnosis of X-linked RP, they were never really convinced, because I guess this is of their son. They were never truly convinced because the birth mother showed insignificant mutations, and there was no one on her side of the family who had RP. They said one distant relative had glaucoma. So how can we be sure that our son's diagnosis is correct, given that his mutations are VUS type? And should we have genetic testing done again, as it's been 4 years?
Okay, so again, throwing up my I am not an MD shield. A couple of things about that is, first off, with X-linked retinitis pigmentosa, females tend to be the carriers of that condition because they have to pass on the X chromosome, like we just talked about. Many times they can be asymptomatic carriers or subclinical, meaning that they don't appear to have the disease but still carry the X chromosome. So just because there's no family history doesn't mean it can't be that. But the fact that you called it a VUS, and a VUS means variant of unknown significance. Sometimes we find a mutation in a gene that we know causes the disease, and we think it might be causative, but we're not sure. It's not something that's come up before. Maybe it's really, really rare, and no studies have been done to decide whether or not this mutation causes disease. This is the absolute challenge with VUSs. These are things that need to be brought into labs using these cell models to look at, does this affect the protein, why and how? And ultimately, I don't have a great answer for you on that one. But if you haven't been tested in 4 years, I would recommend talking to either your medical geneticist or your ophthalmologist about this, because panels are changing really, really fast. You got to keep in mind, I mentioned earlier that medical genetics as a whole is a very young science. We only did complete the Human Genome Project in 2002, and we thought that was going to solve a lot of problems, but in fact, it just raised way more questions than we ever had thought it would. So we're only 18 years into what we could conceivably call medical genetics, and we've made really, really great strides, and things are changing per year. So I would recommend at least talking to your MD.
Great. Okay, so another question I have here from Kim, actually very similar to kind of the same vein you were just talking about. So she says, we understand the vital role identifying a faulty gene can play in the prognosis and potential treatment for retinal dystrophies such as RP. Hoping that you might be able to give some information on treatment options and research developments for those who find themselves in a perpetual limbo, having received an inconclusive result or no mutation found following genetics test. So any work that's being done to sort of help in this area.
Right. So, I mean, as I highlighted in the presentation towards the end, research is slow. This is a reality of research as a whole, because not only do we come upon our own stumbling blocks, but we're trying to educate young scientists as well who are taking on these projects, and it can be slow. And the main thing that we're concerned about is making sure we get it right and making sure our data is. But, and as I said too, the medical genetics is young. We've made a lot of long strides over the last 18 years. I remember reading the paper about Luxturna, which is the newest drug on the newest gene therapy drug on the market now for Leber's congenital amaurosis, or LCA. I remember reading that study in 2005. So within 15 years, they had gone from a concept to a drug in 15 years, which is really good. And it's getting faster as time goes by. If you go to clinicaltrials.gov and have a look at the number of clinical trials involved with gene therapy and with retinal conditions, there are about 35 clinical trials going on right now. And if you look for stem cells and retinal disease, you got about 58, I think it was. I checked earlier this afternoon. So there is a lot of work being done. I can't give any specifics on what is going on right now, but there's a ton of work being done out there. We're getting faster and faster at doing it. And I'm really hopeful for the future. For the next 10 years, I think we're going to see a big transformation in terms of what we can deliver in terms of gene therapies and therapies for these conditions.
For sure. Okay, I'm going to go to the Q&A now. Stephanie asks, she says, I have Best disease, autosomal recessive. I have had genetic testing done twice, but the last time was 15 years ago. Is there value in having the testing done again? I have since had a son. He does not have any vision issues to date, but should I have him tested?
Okay, so autosomal recessive, Best disease, had genetic testing done twice. And I'm assuming if you've had it done more than once, it must have been inconclusive. 15 years ago, I would think, again, hiding behind that I'm not a doctor shield, not a clinician shield, that you should talk to your ophthalmologist or your medical genetics person again to get another test, because if his mutation is in RPE65, there is a therapy in the United States right now which is called Luxturna, which we just talked about. So knowing your genetic status is a really good thing, because it qualifies you for any genetic trials, any clinical trials that might go on, as well as it might qualify you for any therapies that are developed in the next 5 to 10 years. So I would recommend going to get tested again.
Mute. Okay, great. So Rebecca asks, she says, great presentation. Very interested in your work investigating glaucoma caused by FOXC1 mutations. What are the hurdles you're facing in finding an effective treatment for glaucoma caused by FOXC1? And do you have a plan to overcome these hurdles? And then, is FOXC1 a good candidate for gene therapy? Lots of questions.
I love that you've been reading my work, Rebecca. I appreciate that very much. It's so nice to hear that. I figured I was going to publish that and nobody'd read it ever again. But no, again, I appreciate the question. So FOXC1 is a gene that causes a condition called Axenfeld-Rieger syndrome. It affects the front part of the eye, the iris, the cornea, and the lens. And it often causes glaucoma, which is part of the studies that I was doing with Dr. Michael Walter here at the University of Alberta. And what are the hurdles you're facing in finding an effective treatment for glaucoma caused by FOXC1? So the major hurdle is that Axenfeld-Rieger syndrome is a developmental disease, which means that the front structure of the eye hasn't developed correctly. There are molecular reasons, we believe, why it causes glaucoma. And for example, some of my work showed that prostaglandin medications, so some of you might be familiar with like Travatan or latanoprost, Duotrav, these kind of things don't really work well with glaucoma caused by FOXC1. The hurdle there is, like I said, is a developmental disease. So trying to fix the structures at the front of the eye after you've been born is really, really tough and really challenging. And whether or not it's a good candidate for gene therapy is a tough one as well, because when you're doing gene therapy, you're introducing the gene FOXY1 into the environment, right? The problem with it is that we've noticed that too little FOXY1 can cause Axenfeld-Rieger syndrome and glaucoma, but also too much FOXY1 can cause Axenfeld-Rieger syndrome and glaucoma. So it seems like it's on a really, really tight leash in terms of how much it can be turned on that thermostat dial that we talked about. It seems like it's on a very, very tight leash. And so it's also involved with triple negative breast cancers. So you can just imagine the hurdles now. They are just kind of all piled together, and you've got to take one long jump it's not going to happen. So I can't really give you a great answer on any trials that are going on for it right now. I don't believe there are. But yeah, that would be a tough one, I think, to make a gene therapy for.
Okay, great. We're getting near the end, but there are some more questions. I noticed we have a hand up. Sherry has her hand up. So Sherry, I'm going to open up the channel if you want to try to talk. You may have to unmute, and you can do that on your keyboard by pressing Alt-A if you're using Windows, or Shift-Command-A if you're using a Mac. So let's see if we can hear from Sherry.
Hello, can you hear me?
We can.
Yes.
Oh, that's wonderful. Yes, I'm in my mid-80s, and I have two sons in their early 60s. I inherited AMD from my mother. Do you think it would pass to my sons?
So
or does it usually go from mother to daughter, or does it skip sex and go possibly to sons?
Okay, Sherry, thank you for your...
And I also have glaucoma.
Oh, so Sherry, thank you for your question. I really appreciate it. So AMD falls under one of those categories. So we talked about inherited retinal diseases. We talked about autosomal dominant, recessive, and X-linked. Age-related macular degeneration kind of falls under this idea of complex inheritance. There's no set inheritance for age-related macular degeneration. It's not like if your mother has it, you're definitely going to get it. That's called a penetrance issue. If your mother has it, what's the likelihood you're going to get it? But with complex conditions, having a first-degree relative, that is mother, brother, sister, that kind of thing, increases your likelihood of getting it, but it's certainly not guaranteed. I would recommend that your sons get checked regularly by their optometrist or their ophthalmologist to look for signs of AMD. Also, don't smoke and wear protective lenses, because they are at a higher risk of getting it because you have it and your mother had it, but it's not 100%.
Great. That's so exciting. We got an audio question. I never get those to work. That was really great. Thank you, Sherry.
Morgan, can I pick a question to answer?
Absolutely.
Amber Joy Boyd asked, how do you start the conversation to have genetic testing? Who do you ask?
Can I jump in really quick? We have so many questions about this. I have a couple in the chat. So absolutely, how does anyone get genetic testing across the country? Where do they go? What do they do? So
I'm gonna plug the people that are hosting me today and say you guys should absolutely talk to the people here at Fighting Blindness Canada. I'm having a hard time with the Foundation Fighting Blindness and the Fighting Blindness Canada, so it almost always comes out Foundation Fighting Blindness Canada. But anyway, absolutely talk to them. There is the patient registry, which can get you signed up for research projects. And depending on the province that you're in, I'm pretty confident in Alberta that you can get genetic testing covered by going to your MD, and they will refer you to a medical geneticist who can then help you get this genetic testing done. They'll do that initial panel we talked about where they'll look at all the genes that cause the disease that you might have. That may give you an answer. It may not. If it doesn't, then your file might pop up on my desk. Excuse me. What else to add to that? Ultimately, go to your MD and advocate for yourself to get this genetic testing done.
Absolutely. And yeah,
Larissa has some additional.
I'll let Larissa take over there too.
The registry, we always, sorry. I think, am I unmuted now? Okay. Yeah, thanks for plugging our registry. We always like to hear that. So yeah, you can go to our website and find more about the patient registry, or Morgan, maybe you can add that into the follow-up email. Or contact Sherry at the health information line who can give you more information. But for genetic testing, it is now covered in all provinces. So if you get a referral from your appropriate healthcare professional, as Lance said, is covered by every province, which is great news. So, so hopefully that helps you.
Absolutely. And it is covered in every province, but the process in every province, of course, is not the same. So there are these little differences about navigating the system. So definitely feel free to email myself or our health information officer, and we can kind of help you navigate the system, because it is, it's not simple in some provinces anyway. So that was great. So I think that answers a few questions that people had. Let's look at Romolo asked, if stem cells can become anything, at what stage do changes happen? And if genetically these changes are already programmed, how do you eliminate the chance from it happening again in the new injected cells?
Romolo, are you looking for a job? Because that's a really good question. If stem cells can become anything, at what stage do changes happen? I'm guessing you mean in the human body rather than in the dish. Is my guess of what you're asking. And truthfully, I have to back up a little bit here and say I don't know. There are stem cell based therapies going on, or not based therapies, based clinical trials that are going on. And if I can kind of put a little side note on this, there are a lot of people out there offering stem cell based therapies for retinal diseases. Don't buy it. They're offering a lot of false hope, and a lot of people are, not a lot, but some people are getting hurt doing it. So don't buy into anybody who's trying to play on your hopefulness with stem cell based therapies. We're not quite there yet. If genetically these changes are already programmed, how do you eliminate the chance from happening again in the new injections? And I guess you're saying, how are that they're prevented from being turned back?
Yeah, I think he means like, how do you prevent the disease from just like expressing itself again in the cells?
So the new injected cells would likely come from a donor who would not have the same genetic mutation that you would. So ideally, this is the same as like getting a heart transplant, right? The heart comes with its own set of genetics. Same with that with the injected stem cells, would have its own set of genetics, which would not be yours, but it would have to be matched as well to make sure that you won't go through a rejection or anything like this as well. So I guess that's probably the best answer I can give you, Romolo, but that's a really, really great question.
Great. So we have a couple questions, one in the chat from Shaman and one from Kim. And I think they're kind of the same, basically asking like, what is the next step if someone has been diagnosed with a retinal dystrophy, their genetics tests aren't showing sort of mutation? Is there more thorough genetics tests that can be done? And Shaman was asking like, how do we get you to have a look at their file? Like, how does it get to you?
Okay, so for Kim, mutation identified. Are there more thorough genetic tests? So the one I talked about is whole exome sequencing. That just looks at that 2% of the genome that I talked about, just the genes. The next step up is called whole genome sequencing. That takes care of 100% of the genome, that 98% intergenic, the thermostat regions, and the 2% of the genes. That is the best way to go about looking for the new mutation, but that is typically done on a research basis. I don't think that's offered clinically. Again, standing behind my not a clinician shield. But I would think that would be the best way to go about it. And as how to get your files to me or people like me who do the research is, again, you need to talk to your doctors, your MDs, tell them about your family history. Particularly if you can get a referral, I think, to a medical geneticist, that would be the most valuable thing, because they can help determine whether or not you'd be eligible for testing. They help you determine if you'd be eligible for research, and maybe give some good advice on where to go from there. And then from there, you can also get genetic counseling.
Great. I know we're just a little over time. We have one more question and then we'll wrap up if that's okay. So I have an email from, so there is a couple questions in the Q&A, but if we don't get to you today, we'll send you some information over email as well. I will follow up with you for sure. So Margaret says, I'm 76 and developed dry and wet AMD 6 years ago. She says she's managing very well with frequent intraocular injections and Vitalux. She's able to drive and read. She said her mother had the disease and went blind before anti-VEGF treatment. Her sister did not have AMD, but had poor vision and chronic iritis. She has a son and daughter who are 48 and 50. Neither of them seem to have vision problems right now, except they're starting to need glasses. So what are the chances that they're going to develop AMD? Her husband doesn't have AMD, but has glaucoma. And are there preventative measures her kids can take now to sort of prevent any onset of disease?
Okay, so I'm going to fall back on the same answer that I gave earlier, where so AMD is not necessarily one of these inherited retinal dystrophies. It's one of the complex conditions where we don't really understand exactly which genetic variations are playing a role here, but we know that some contribute a higher risk of you developing AMD. There's also environmental factors, like smoking is definitely a higher AMD factor. There is some suggestion that UV light might play a role. So I guess the best advice I could give for your children are frequent checkups to make sure that their eyes are still healthy. Don't smoke. If they smoke now, quit. And wear protective eyewear when outside to minimize exposure. That last one is not 100% true, I don't think. I think the science is kind of up in the air on whether or not UV light actually contributes to progression of macular degeneration. But I think better safe than sorry in these cases. So yeah, so what did I say about that? Yeah, and like I noted earlier, anytime you have a first degree relative who has a complex condition like glaucoma, like AMD, these increase your risk of developing it, but it's not 100% risk, right? If your chance of developing it was 1% before and you've increased your risk by 100%, that means your chance is now 2%, right? So you got to keep that in mind. So it's something to look out for, frequent checkups, some preventative measures, but ultimately, yeah.
Great, thank you. So yeah, there were a few extra questions, but I will be sure to reach out to you with some more information. Thank you to everyone for asking great questions today. And thank you, Dr. Doucette, for joining us. We met last year in Alberta for the first time, and he spoke at Vision Quest, and I was immediately struck by how well you're able to kind of explain these very complicated concepts so that I could understand them.
Appreciate that.
It's great, and thank you so much.